Matariki is the Maori name for the star cluster Pleiades, also known as the Seven Sisters. Matariki is also the name for an international network of 7 universities that are distinguished by their historic traditions and unique approach to facing contemporary challenges. The Matariki Lecture Series offers a short program of lectures addressing current research themes or areas of common interest. This time, the Matariki Lecture Series is on a topic of human evolution.
We all know some basic claims of the evolution of mankind, but, and this is the question we want to address today, is the human evolution more complicated than we first thought? With these introductory words, a warm welcome to everybody. A warm welcome to the Matariki Lecture Series 2021. The topic today is human evolution. My name is Oliver. I'm the head of Campus TV, the student television of the University of Tübingen, and I'm hosting the event today, and I'm happy about this. With me in the studio is Madeline Böhme from the University of Tübingen. On screen, connected by Zoom, is Per Alberg from Uppsala University, Jerry De Silva from Dartmouth College, and Sarah Elton from Durham University. Those are our 4 speakers. On screen too is Monique Scher. Monique Scher is the Vice President for International Affairs and Diversity of the University of Tübingen, and she will give some warm words to us too. Monique, the stage is yours.
Thank you, Oliver. Good afternoon, everyone. It's wonderful to see the faces that I can see on my screen. And I hope that there are a lot of faces out there watching us today. As Oliver mentioned, I'm Monique Scher. I'm the Vice Rector of International Affairs and Diversity at the University of Tübingen, and I've been involved in the Matariki Network, therefore, as in my role as Vice Rector. The Matariki Lecture itself is a format we started several years ago, and it began as an annual event. A faculty member from one of the partners would deliver a public lecture at an institution, another institution within Matariki, as part of a broader visit to the campus. The first lecture took place at Queen's University in Canada and was given by a researcher from Dartmouth, for example. And in 2018, it was in Tübingen, a lecture from Durham. In 2019, in Otago, someone spoke from UWA. And so it sort of made the rounds that way. But this lecture in 2020, of course, we had to start thinking about making it virtual. And now the pandemic has forced us to sort of rethink the format altogether. Free from the constraints of physical travel, we've decided to turn it into a series. And a series of lectures in short progression, that would be an exciting format to use and to showcase the research in our network. So this new format here today is the beginning of this new lecture series, the virtual Matariki Lecture Series. And it starts with this exciting question Oliver mentioned: is human evolution even more complicated than we thought? And we will hear presentations by scientists from the 4 partners that were mentioned: Dartmouth, Durham, Uppsala, and Tübingen. Beginning with Professor Madeline Böhme from Tübingen, and moderated with a question and answer afterwards. So it's an excellent opportunity for anyone listening to ask the researchers, all of them at the cutting edge of their fields, from 4 different countries, to ask them anything you wanted to know about human evolution in this new Matariki virtual exchange format. So again, welcome everyone. We all look forward to an exciting set of talks, and back to you, Oliver.
Thank you very much, Monique, for your warm words. Before we start, I have a few words on the procedure today. We have 4 lectures. Each lecture takes about 15 minutes. And after the lecture, we have a Q&A session. You can pose your questions, and we're gonna give, not me, but all the speakers are gonna give the answers. We have a Q&A window by Zoom, and when the first lecture starts, you can already pose your questions. Because we have somebody sitting in the back, he's collecting the question, he's sorting them, and he's sending the questions to me in the studio. So don't hesitate to pose your question when you have your questions, even though a lecture is running. So, I think we're going to start. We start with Madeline Böhme. Madeline Böhme is from the University of Tübingen. Madeline Böhme is doing research in geoscience and paleontology and paleoclimatology. Her research looks at the human evolution in relation to changes in climate and environment. The title of her lecture is Danuvius Guggenmosi and the Positional Behavior on the Last Common Ancestor of Great Apes and Humans. Madeleine, the stage is yours.
Thank you, Oliver. Yeah, hello everybody. It's a pleasure for me to be in this very first Matariki lecture of this year. Yeah, my talk is about Danuvius guggenmosi. It's a new ape species from the late Miocene, long ago, from Germany. And what this species can tell us about positional behavior of the last common ancestor of great apes and humans. And since the overarching question of our lecture today is human evolution more complicated? I would say yes, it is. And maybe you all know or have remember a similar cartoon like this. This is the original cartoon published in 1965 by Clark Howell in his famous book of Early Man. And what you see here is the ascending order of human evolution. And you see that the human evolution was imagined at this time as a series of events becoming more and more upright. And especially if you look on the left side, then several creations you see there which are looking a little bit gibbon-like, some are looking chimp-like. And the problem is that all these creations on the left side have been in the 60s largely in imagination, because the fossil record have been not able to tell us anything about the positional behavior of these very old Miocene species. And so the paradigma is that the anatomy of our most nearest living relatives, the great apes, is primitive. And that means that the last common ancestor has also a quite primitive anatomy. And you see these both pictures of a gorilla and of an early Miocene ape that we imagine, the paradigma imagine, the last common ancestor as having bent hips, bent knees. So a little bit like a knuckle-walking living great ape. And the living great apes are indeed because of genetic data, our nearest living relatives. And they are knuckle walkers. But the question is our last common ancestor a knuckle walker? Or, in other words, are chimps and gorillas primitive in positional behavior? And we, as bipedal humans, are advanced? So this is a question which only can be answered by fossil record and not by theory. So the fossils are our base. And the new fossil, Danuvius guggenmosi from the late Miocene of Germany, is exceptional because we have a lot of parts of the body, and we can tell from nearly every or many important joints in the skeleton something about positional behavior. And my talk will be about these things. And the main result seems to be that bipedalism, which is characterizing us humans, hominins, maybe originate in arboreal settings, and maybe quite early, more than 12 million years ago, and maybe in Europe. So this I will demonstrate. If you are looking on the anatomy of a modern human and a chimp, for instance, then we see several similarities, but we see also a lot of differences. And they are related to the bipedalism of humans on one side and to the quadrupedalism and knuckle walking and suspensory behavior, that means below branch hanging behavior of great apes. And you see several words on the left side and on the right side which are documented from Danuvius and which show that Danuvius is a mosaic of several anatomical parts characteristic for bipedalism and for suspension. And I want to start with the suspension characters. So, for instance, in Danuvius, the arms are longer than the legs. So the ulna is about 18% longer than the shin bone, the tibia, which is a typical number for chimps, for instance. The phalanges of the hands are curved, which is also a very typical anatomy for suspensory apes, and in a similar way a chimp phalanx would look like. But very interestingly, a halocal phalanx is also preserved, and this phalanx is not only very large, extremely large and robust, it has a torque to the lateral side. And this is a very important indicator for a grasping foot. And these grasping foot abilities of Danuvius are probably much stronger than in chimps or in gorillas today. So the foot or the hallux and the hand and the length of the arms indicate a suspensory behavior in Danuvius. But on the other side, there are several parts of the body which speaks for a more bipedal part of his life. And I want to give you some examples. You know all that humans have a spinal lordosis. So the spine is curved in contrast to great apes. And if we are looking on the thoracal vertebra of a chimp, then you observe that the dorsal spinae have an inclination which are all the same along the spine. And if we are looking on humans, you see that the lower part of the thorax vertebra have a higher inclination. And this don't look very comfortable here. And the reason is that in reality, humans have a lordosis, so have a curved spine. And luckily for us, we have two thoracic vertebrae from Danuvius. The very first one, the very upper one, and the one from the middle of the body. And you see the inclination is quite different. So the inclination difference is such high as it is in modern humans. This is an indication that the spine of Danuvius has a lordosis similar to bipedal humans or hominins. If you look on the thorax itself, then you see in the upper left a chimp. A chimp has a very narrow but very deep thorax. In contrast, humans, we have a very broad but a very flat thorax. And this you can analyze in the fossils or in vertebrae if you are looking on the first vertebra of the thorax. And you see below of living great apes, of orangutan, a gorilla and a pan, the transversal processes are horizontal. And this means that the vertebrae are not invaginated in the thorax. In humans and in Danuvius, you see that the transversal processes are dorsally orientated. That means the centra of the vertebra are invaginated into the thorax, which is a feature we find today only, or we find only in hominins, so in fossil and extant humans and nearest relatives to them. If you are looking on the vertebrae itself, on the poor numbers of vertebrae, humans and great apes don't differ much. But there's a big difference. In humans, there are longer or more lumbar vertebrae. So the lumbar spine is longer because it has to be flexible. So it has to be flexible to have the heavy body over the hips. In great apes, there is only a short lumbar vertebra, with only three vertebrae, and they are stiff. And this stiffness is very important for the apes for climbing. If you are looking on the nuvius, we have one vertebra from the transition between lumbar and thoracic vertebra. And this is a transitional vertebra or a diaphragmatic vertebra. And the position of this transitional vertebra in modern humans, you see on the right side. The red color is the number 12 of the thoracic vertebra. So it's at the end of the thorax, the lower end. In the nuvius, this transitional or diaphragmatic vertebra is higher up within the thorax. That means that the lumbar spine is quite elongated. Elongated means more than 4, 5, maybe 6 or 7 vertebrae, similar to Australopithecus or other early humans. An indication for a flexible lumbar spine. If you are going now to the legs, especially to the knees, then there is a very important difference between humans, bipedal hominins, and apes. That is the so-called valgus knee for bipedal animals. That means that the knee is positioned directly below the center of gravity, so below the hip and not sidewards to the hip. And to understand this condition in the nuvius, you need a shin bone, a tibia, and we have a tibia. And you see here a cast of the tibia of the nuvius, and you see that the ankle joint is perpendicular to the long axis of the tibia, so it's 90 degrees, more or less. And in chimps and other great apes, there is a higher angle. So that means the perpendicular orientation of the long axis of the knee to the ankle means that the nuvius had a valgus knee, that the knees are directly below the midpoint of gravity. Another indication for a bipedal posture. If you are looking now on the knee itself, then there is a big difference between bipedal humans and relatives and great apes. And this is on the plateau, the tibial plateau, where the tibia, or the shin bone, and the femur are in contact. In humans, this surface is flat, absolutely flat, and this is important because we stand with extended knees. That means for us it's no problem for hours to stand upright with extended knees. And that means that the femur and the tibia should have a broad contact, a comfortable broad contact. And the same condition we find in the nuvius. So the plateau is flat. But if you're looking in great apes, you see this plateau is curved. And I show you here a gorilla, and it's extreme variant of curved. And this means that a gorilla is more happy to have flexed knees. So that means extended knees is a very uncomfortable situation for great apes. And I did this with zoo animals, and they don't want to have extended knees. They go immediately in this condition. So this is again a bipedal posture indication for the nuvius. And last, at the end of these examples, I want to show you something about the tibiota joint. And in humans, this joint is quite stable because we are adapted to a bipedal walking and running, of course. And in apes, it's a quite flexible joint because of this needs to climb and to hyperflex the foot. And indeed, if you compare this joint from a chimpanzee with the nuvius, you see that the chimpanzee and the great apes in general have a trapezoid contact. That means the anterior part is much more broad, and the medial malleus is a very strong bone. In humans and in the nuvius, the surface is more rectangular, and the malleolus is very thin or relatively thin. Again, an indication that this joint in the nuvius is more human-like than great ape or living great ape-like. Okay, so that means we have a mosaic of different anatomies And we cannot compare this with a living animal, with a living primate. So we, in our publication about Danuvius, we defined a new positional behavior type, which we named extended limb clambering. You can also say kind of arboreal bipedalism. So this positional behavior combines suspensory climbing abilities with slow and careful bipedal walks on narrow arboreal support. And these pictures here give you an imagination, or at least how we at the moment can imagine the feet and the positional behavior of Danuvius. So coming back to our initial question, how complicated is the evolution of humans and great apes? I would say it's now getting more and more complicated. We don't know exactly the phylogenetic position of Danuvius really at the moment, but what we see is that probably the anatomy of living great apes is not such primitive. So they are probably quite advanced and well adapted to a present-day environment, which is in the rainforests of Africa or in Southeast Asia. So, and that the positional behavior of African great apes, the knuckle walking, is probably a homoplasy. So it evolves several times, it is advanced, highly advanced, and is not primitive. And some features of human bipedalism may be indeed more primitive than we have thought before. So these are new questions arising from new fossils. Thank you for your attention.
Thank you very much, Madeline, for your exciting talk. I'm sure we're going to have a lot of questions later on in our Q&A session. The next speaker is Per Ahlberg. Per Ahlberg is from Uppsala University, and Per is doing research on vertebrate evolution. The research is ranging from short vertebrates to certain aspects of later vertebrate evolution, including the origin of hominins. Per Ahlberg focuses on trace fossils and body fossils as data source for understanding vertebrate evolution. Per, we are all looking forward to your talk. The title of the talk, the Trachelos footprints, what are they, and what do they tell us about early hominins?
Right, yeah, I couldn't remember exactly what I'd called it, so I'm afraid I changed the title slightly, but it's the same content.
No problem.
Thank you. Can you all see the screen?
Yes.
This is working? Okay, super stuff. Then I'll run along. Well, this follows on very, very neatly from Madeline's talk, and I will try to confuse you further about human evolution, turning now from the body fossil record to a complementary source of data, namely the trace fossil record. Fossil footprints of land vertebrates, and in fact, fossil footprints of all sorts of things. You can look at, millipedes or insects if you're interested in that kind of thing. But if we're thinking vertebrates, fossil footprints of land vertebrates, of tetrapods, are relatively common through the entire record of the group. The earliest tetrapod footprints that we have in the fossil record are rather more than 380 million years old. So they're relatively common. You see a few examples here. There's an early reptile on the left, dinosaur in the middle, and of course the famous Laetoli prints of an early hominin on the right. They can be very informative about foot morphology, locomotion, and indeed behavior. Interestingly though, footprints of early hominins are pretty rare. What you're seeing here, the Laetoli prints, are the oldest kind of universally accepted hominin prints. They're 3.66 million years old, which sounds kind of impressive, but recently calibrated molecular dates for the human-chimp split tell us that it likely happened over the order of just shy of 8 million years ago. So that, in other words, if you take the entirety of the hominin lineage from its separation from the chimp lineage up to us lot sitting here today, the Laetoli prints are more than halfway along that evolutionary trajectory, which is quite remarkable when you think about it. What on earth were we actually doing during the missing 4.24, or whatever it's going to be, million years? Well, I think I might be able to show you what we were doing. I'm going to be telling you about a footprint locality in Western Crete, originally discovered by Georg Gerlinski and described by him and myself and a bunch of other colleagues in a paper in 2017. We originally dated this locality at the time rather loosely to sort of ballpark 5.7 million years old. This is on the basis of a combination of stratigraphic data and microfossils. It's recently been redated. We have a manuscript kind of in the pipeline in this, redated now to 6.1 million years, so just a little bit older. Very important thing to appreciate, you know, Crete's a lovely spot, but back in those days, it was not an island. We're not talking about some sort of, you know, weird island biota here. Crete at that time was a peninsula projecting from the Greek mainland. In other words, it was the southernmost tip of the Balkans. Okay. You're probably already aware that these, or you soon will be otherwise, that these footprints have been the subject of considerable controversy. And some people have wanted to claim that they're in fact not footprints at all, but just kind of like, you know, holes in the rock that look a bit like feet, which of course would rather puncture my talk. I just want to kind of put down a marker here from the beginning. These are perfectly obvious and indeed rather well-preserved footprints. They have a whole run of characteristic features that you only see in footprints, not in generic holes in the ground, such as expulsion rims. Can you see my cursor moving, by the way? Excellent. So these are the little sort of squish rims that happen when a foot pushes down into the sediment and the sediment is squeezed out. Also these somewhat peculiar, what we call pull-up features, which is the sort of thing that happens if you're walking on a rather sticky, perhaps calcareous mud, that sort of thing. It'll sort of go swap like that when you pull your foot up. So suffice it to say that there's really no question whether these are absolutely real footprints. Oh yes, we also have toenail drags on some of them. And I just want to kind of mention in passing here that the author group that described these have decades of experience of footprints, and between them more than 200 peer-reviewed papers on footprints. So they're footprints, okay? Right, rolling on then, if they are footprints, who made them? Well, that's an interesting question. They appear to be made by a bipedal animal because we only find one kind of foot morphology in the prints. We have a few definable trackways. A lot of it just seems to be kind of milling around, to be honest. But there are a few trackways which are notably narrow and linear. This, of course, tells you you're bringing your feet in close together. CF, what Madeline was just saying here about knee morphology and so on in Danuvius, these points are worth thinking about. You're probably looking here at an animal with a valgus knee, or it wouldn't really be able to make this kind of narrow single line track. What you see here in gray is a laser scanned surface of the principal footprint surface, which, by the way, is some sort of, you know, wet beach sand, probably a little sandbar in a very small river estuary, that kind of thing, on the north shore of this little peninsula. So we're going to look at the three best preserved footprints, and they look like this. And what I think should be immediately striking to you is that they have a sort of human-like gestalt to them. They're shallow, but the details are often quite crisp, and we can see either 5 or 4, depending on the preservation of the print, digits. So it's a distinctively human-like morphology. And of course, in case you weren't aware of this, these kinds of feet are actually unique to hominins. I'm rather fond of this image. This is from an old 19th century edition of Robinson Crusoe I have at home with those lovely wood block engravings. And here, of course, is that moment when Robinson, thinking himself all alone on his island, discovers to his horror a human footprint on the beach. The point is, of course, that what everybody realizes, what Defoe realized when he wrote this scene, and what the illustrator realized, is that a human footprint is utterly characteristic. Robinson isn't going to be standing there wondering whether this is like, a goat or something that's wandered by. In fact, in the extant biota, the human foot morphology, the human style of bipedal walking is absolutely unique. And even if we look back in time along the hominin lineage, we don't really find feet like this in anything else. So this is immediately very interesting. But these feet, and that makes them even more interesting, are not entirely human-like. Let's have a look at this a bit more carefully. I'll zoom in on this, which I think is perhaps the most informative of the prints. If we look at the toes and the front part of the sole, they are remarkably human-like. We've got a big hallux, we've got smaller lateral digits, we've got a ball with a sort of, subpart here behind, kind of a distinct big toe ball, very much like you would see in a modern human foot. There are differences, but they are really very subtle. If you look at the back end, it's not nearly as obvious. You'll see immediately that this part of the foot is proportionately a great deal shorter than in the human foot. Also, the modern human heel is bulbous, whereas the heel here is rather narrow and pointed, giving a sort of heart shape look to the whole thing. And there's no real arch. Of course, we have a strongly developed medial arch in the modern human foot. You find when you look at these that there's a certain sense that the animal is putting more weight on the lateral edge of its foot. That's probably why we're getting the pull-up feature here. But there's not a lot in it. Certainly, you can't really point to a distinct arch being present. What do we make of that then? Well, I mean, the human-like bits are human-like. The other bits fit with what one could think of as a rather generic primate sort of look. And this, of course, is a character combination that immediately begins to suggest that we may be looking here at a very primitive hominin. Not quite a human, certainly it's not quite a human foot, but something that is in the process of assembling the human foot character complement. Right, well, we had a bit of a time trying to get this paper published. It took us a number of years, and we had some very, very hostile responses from the paleoanthropological community. The idea of a Miocene hominin in Europe clearly wasn't going down well at all. And of course, one issue that was frequently raised, and indeed should be raised as a very legitimate question, is, well, could these have been made by something else? Assuming they are footprints, what's telling you that this is some sort of basal hominin? And the two obvious candidates that came up again and again were bears on the one hand and apes on the other. I particularly like this. This, of course, is a gorilla in captivity. I forget where this guy lives now, but he's presumably, by looking at humans and thinking this is a great idea, is adopted bipedal walking and is just kind of freaking out the visitors to his zoo. All right, could it be a bear? No, it could not be a bear. Why do I say that? Well, when you look at bear prints, you see some very distinctive differences. First, you can notice with this bear standing up right here, bears have got socking great claws on both forelimbs and hind limbs. You never see claw impressions in any of the Trachelos footprints. There's not a single trace of them. So I think we can safely conclude that this is not some sort of preservational issue, but that these things simply did not have claws. And in fact, you can see rounded tips of the toes, and the lateral toes is having these rather square cut ends, very much like the lesser digits in a human foot. Also, when you look at the digits of a barefoot, bears walk barefoot, Anyhow, enough of that. The digits are all essentially similar in shape. The impressions all look like imprints of olives, basically, of slightly different sizes. And that's not the case here. You have this obviously distinctive hallux, which has a narrow neck, very much like in us. And you can see, although it's extremely faint here, that plainly the lateral digits are rather sort of parallel-sided and a quite different shape. And again, the sole is just a different shape. So all in all, I think we can dismiss bears quite happily. These are not a good fit for bear prints. What about some sort of ape then? Perhaps a more reasonable suggestion. Well, if you look at the feet of modern apes, you find that all of them have feet that look rather like hands. They all have divergent halluces. So there's a big gap between the big toe and the other toes. And this, of course, relates to the fact that they all retain a grasping function in the foot. And this, of course, also comes through in their footprints. These aren't, well, that's a nice photograph. This one's kind of rubbish. But the reason why I wanted to use those particular ones is I'm always a little careful about footprints that are made by zoo animals. A zoo is not a natural setting. You always wonder a little bit whether in some way the behavior of the animal could be affected. This is a footprint of a wild bonobo, and here's a footprint of a wild gorilla, in fact, with a lovely knuckle print next to it. So this is what they do out in the bushes. And you can easily see that what's characteristic of these footprints is that they are Y-shaped. You always have a substantial divergence between digits 1 and 2. And it is equally clear. Also, the other thing is, if you think of the lateral digits here as forming sort of a line, and you think of the edge of the sole as being out here, you can see that the hallux is set back along the side of the sole, like our thumb is set back along the side of the palm of our hand. When you look at the trachelous footprints, they're not like that. There is never a significant gap between digits 1 and 2. And what's more, you can see that digit 1 lies alongside digit 2, both of them springing from the distal margin of the sole. It's worth spending here a couple of minutes on the one really serious rebuttal of our paper that's been published as a comment in the same journal by Meldrum and Sarmiento the year after our piece came out. This is a rather peculiar sort of article because you could boil it down, and I am not misrepresenting them here. You could really boil down their argument to saying, well, these are not footprints. But if they are footprints, then they are ape prints. Now, for me, that doesn't really add up. But if we sort of roll with this, this is one of their actual comparative figures to which I have added the nices of the trachelous feet. And they put this up with the purpose of showing how deceptive a gorilla footprint in the middle here can be. So here's a gorilla walking with his big toe pulled in. And they say, look how similar it is. And look how like the trachelous footprints they are. And then they put in a Laetoli foot here for comparison. But this doesn't work awfully well because you see, if you look at this, you find here, obviously, this is now a human. Digits 1 and 2 close together and they're level. Here in the gorilla, digit 1 is, not only is there still quite a gap, but digit 1 is visibly set back from digit 2. In the trachelous footprints, they're really tight together and they are side by side. Also, of course, the sole margin is convex in the human, reflecting the presence here of the ball. He has this most peculiar transverse, sort of concave transverse margin, and the margins in the trachelous footprints is convex. So actually it shows the exact opposite of what Meldrum and Sarmiento wanted to demonstrate. So there you go, these things happen. I can also add that a really very nice morphometric analysis of the trachelous footprints in our paper, this made by Matthew Bennett, who really is pretty much the leading figure in this kind of analysis of hominin footprints, showed very clearly that the trachelous prints, you have them, so the usual principal components analysis thing, components 1 and 2, components 1 and 3. You can see that the trachelous footprints here in gray cluster tightly with the Laetoli prints and modern human prints well away from non-human primates, and including in the little sort of blue outline here, triangle-shaped baboons, which are, of course, kind of interesting because they're habitually terrestrial. So this is an unambiguous hominin signal. I would have to say by now that the evidence for a hominin track maker identity for the trachelous footprints is multifaceted, internally consistent, and to my mind appears conclusive. Anybody wants to argue with that, they're very welcome, but I'm not quite sure that I can see where this is going wrong, if it is. So I'm going to roll on with the assumption that these are hominin prints and consider for a moment what they might actually tell us. Well, there's the biogeographical thing, because it stretches the hominin distribution in the Miocene up into the Balkans. But to my mind, that's not the most interesting aspect of this. It's, some people get very hot under the collar about this. For me, this is a minor point of biogeography. What's much more interesting is what they tell us about human foot evolution. This is a figure from our 2017 paper. And at that point, as I mentioned, the dating of the footprints was still a little bit vague. We can now put it more securely there. And the approximate age of the human chimp split is, around here somewhere, a bit further back in time. So you can see that the trachelous footprints kick in about level with the earliest parts of the hominin body fossil record, especially now that Sahelanthropus is looking a little bit shaky. Aurorin here seems on good grounds to be, bipedal, primitive hominin. The comparison between this and the trachelous track maker potentially very interesting, but there is as yet really rather non-overlapping data sets. But they're basically contemporary animals, and I would certainly want to understand more in the future about how these things relate to each other. What becomes a bit trickier is Ardi. So Ardipithecus ramidus then, considerably younger at about 4 and a bit million years. supposedly upright, supposedly a hominin, supposedly, and has been claimed to be, you know, very much in the line towards later hominins, but it's got ape-like feet. It certainly has too. Foot's nicely preserved and, you know, without that correctly interpreted here, nice divergent hallux. Obviously nothing like this at all. So what are we to make of that? Well, of course, I mean, as Madeline has already flagged, we should not think of hominin evolution as a sequence of gradually more upright and less bestial ape men. That's not, you know, nothing evolves like that. It just doesn't work like that. We're thinking here about a radiation of a smallish mammalian clade. And there's every reason to think that this was a multiply branching process. In fact, we know from later in hominin history that that is the case. And there's nothing implausible about there being a deep branching lineage kicking around for a few million years and eventually manifesting itself visibly in the form of Ardipithecus, a lineage that branched off before this, one presumes, terrestrial and no longer very grasping at all sort of foot morphology had become established. Alternatively, conceivably, Ardi is simply not a hominin, but that's really, I think, a debate for another day. Finally, making a quick comparison here with the Laetoli prints, there are obvious similarities between them. The comparison is made a bit more challenging by the very different substrates. The Laetoli hominins were walking in this soft, yielding volcanic ash, producing very three-dimensional prints. Here you're looking at wet, compact beach sand. And of course, a footprint is not a static, like, representation, a cast of a foot. It's a dynamic thing produced the interaction between the foot and the sediment. And so you're going to get differences emerging from these, no doubt, different interactions. I mean, this curious curling ridge here, for example, this is a laser scan of one of the Laetoli prints. I mean, this is clearly not a piece of primary morphology. It's something that is produced by the morphology interacting with the sediment. What we can say, however, is that once again, the Laetoli prints lack an obvious arch, have a kind of pointy heel, but they're longer than the Tachylos prints, and to that extent look considerably more human-like. So a reasonable guess here is that here we are looking at a more crownward hominin, if we put it that way. We're obviously within the hominin stem group here, a more derived animal, more closely related to you and me than this poor thing. But I'll be very interested to hear more about the, these of course have been somewhat loosely attributed to perhaps being made by Australopithecus afarensis, but that wasn't the only hominin around at the time, and I should be very interested to hear more about Australopithecine foot diversity later on. So what conclusions can we draw then? Well, the Tachylos footprints are of the order of 6.1 million years old and were made in a mainland setting on the southernmost tip of the Balkans. They were made by a bipedal animal that lacked claws. The anterior part of its foot was remarkably human-like, with a large and fully adducted hallux, progressively smaller lateral digits, and a transverse ball. But the posterior part of the sole was less human-like. It was short, heart-shaped with a narrow heel, and there was at most an incipient medial arch. So this character combination strongly suggests that the track maker was a very primitive, but bipedal, and indeed probably valgus need hominin. So if we buy that interpretation, then the age of the track maker, and of course this also ties in with the skeletal morphology of Aurorin, shows that bipedality evolved early in the hominin lineage. Well, of course, Madeline's just knocked this out of the park. Bipedality might go right back into the common stem group of hominins and the African apes, but at any rate, bipedality seems to be around a little more than 6 million years ago, suggesting that the ape-like foot of the later Ardipithecus indicates either that that is a very deep branching hominin or possibly that it's not a hominin at all. The Laetoli track maker has a more human-like foot morphology than the Tachylos track maker with a longer sole and was presumably a more derived, more advanced hominin. That's really all I have to say. I've got a whole bunch of people to thank listed here. Gerd Jelinski, obviously, my main collaborator in this, Grzegorz Niedźwiecki, Matthew Bennett for the morphological analysis, Pabis Fasolas and Athanasios Athanasiou for their constant support of the project, the entire Burma team, the organizers, thank you very much for inviting me to this, and the Knut and Alice Wallenberg Foundation who fund my research. And thank you all for listening.
Thank you very much, Pierre, for your interesting talk on Tachylos footprint. I think the next speaker is perfectly connecting to your speech. The next speaker is Jerry. Jerry is from Dartmouth College, and Jerry is doing research in the field of paleoanthropology, and Jerry's research focuses on the locomotion of the first apes and the early human ancestors. His expertise is the anatomy of human foot and the anatomy of human ankle, contributes to the understanding of evolution of upright walking in the human lineage. The title of the talk, All Walks of Life, Bipedal Diversity in Australopithecus. Jerry?
Thank you very much. Yeah, can you hear me okay?
Okay.
Yeah. Fabulous. And everything look good on the screen?
Great.
Great. All right. Thank you so much for having me. I really appreciate all the work that the organizers have done to put this together. And to my fellow contributors, Madeline and Per, I think what I'm going to talk about really nicely transitions from what you talked about. And in fact, Per, we were at Laetoli in 2019, and we made some really fun discoveries that I can't wait to be able to talk about. We're not quite there yet, but I think it nicely complements what you've been talking about. And so I'm gonna shift things to Africa, and I'm gonna shift things a little forward in time and talk about bipedal, not bipedal origins, but what happens after bipedalism is established, and what do we see in terms of the pattern of bipedal evolution throughout what we would call the Pleistocene, and maybe even the late Miocene. And so to contextualize this, just for the listeners that are familiar with human evolution and need sort of a bigger picture, humans are, of course, related to the African great apes. We are first cousins of chimpanzees and bonobos. And as has already been discussed, we share a common ancestor with them that lived somewhere in the ballpark of 6 to 10 million years ago. This was a messy split, though. And so you'll see these different divergent states. And most folks would have the clean separation of the lineage heading to humans and the lineage heading to chimpanzees by 6 to 7 billion years ago or so. And what do we know about the fossil record from this time period, at least in Africa? Some of the earliest fossils that have already been discussed from about 4 to 6 million years ago are best represented by the partial skeleton just discussed, Ardipithecus ramidus, from the Aramis region of the Afar regional state in Ethiopia. And this is a very curious creature. It's got a brain about the size of a chimpanzee brain, relatively long arms, long curved fingers, and that grasping big toe. So this is an animal very comfortable in the trees, but there are other aspects of its foot and of its pelvis that might suggest that it could also move on 2 legs when it was on the ground. It certainly didn't knuckle walk. It doesn't have any of the features of a knuckle walker. By 4 million years ago, though, we start to see a different kind of animal in the landscape. And this is what we call Australopithecus, represented here by the famous Lucy skeleton. Many different kinds of Australopithecus that lived, in fact, until about 1 million years ago, at least in South Africa. And there's another group of them, a cousin of them, that has really big cheek teeth that we sometimes call robust Australopithecus or Paranthropus. Their brains were slightly larger than in Ardipithecus. They had gorilla-sized brains in chimpanzee-sized bodies. And from the neck down, they're much more human-like and show evidence of established obligate terrestrial bipedalism by Lucy's time. There's also evidence of the beginnings of stone tool construction in Australopithecus, although this is a little more controversial at a site in Kenya going back about 3.3 million years ago. By 2 million years ago, we start to find members of the genus Homo, and these are identified as hominins that had longer legs, larger brains, smaller cheek teeth, a greater reliance on technology for survival. So we find more advanced technology, even the invention of fire in sites that are 1 million to 1.5 million years old. They also begin to expand their territories, expand their ranges, and we find them outside of Africa as well. By 1.5 million years ago, they're as far west as Spain and as far east as Indonesia. Back in Africa, however, we have fossil and genetic evidence for the origins and evolution of our own species, not at any one place and time, but instead over a long time span of 200 to 300,000 years ago, throughout the African continent, a pan-African event of gene exchange leading to the evolution of the anatomical characters that we recognize in modern Homo sapiens, modern humans today. But not only do we recognize these anatomies, we recognize these behaviors of symbolic representation of thought and artwork and transport of materials over long distances and all the great things that make us human. But as Madeline discussed, and as Per discussed, and as I'm going to discuss, what do we see way, way, way back? What's the first thing that really distinguishes us from other primates and our ape cousins is this unusual form of locomotion, the ability to move on two legs, bipedalism. And so bipedalism is a hallmark of being human, as was discussed a moment ago with those fossil footprints. Those footprints at Laetoli, as Per said, are unmistakable. You see footprints like that on a landscape, and you know a human or hominin has been there. And of course, we've even left these amazing footprints on another world. And so to me, one of the great places to study bipedalism is the thing in contact with the ground, and that's the foot. And so I study the fossil evidence of foot evolution throughout ape and hominin evolutionary histories. Humans have on the surface feet that look quite different from, say, chimpanzees, although one of the things that I'll emphasize is that our foot is composed of the same 26 bones that a chimpanzee's foot is composed of as well. They're just these subtle modifications in those bones that allow a chimpanzee to do what it can do with its foot and allow us to do what we can do with our feet. Now, to really understand this, though, we have to look at the fossils. How did the foot evolve? And if you go back to sort of early 20th century comparative anatomists who were thinking about this and grappling with foot evolution, they didn't have much to work with. They had Neanderthal feet and modern human feet, and comparisons were made between those. But fortunately, in the 1960s and 1970s, as work was done at Olduvai Gorge and at the site of Hadar in Ethiopia, there were the discoveries of fossilized foot bones, and they were framed, they were interpreted in this context that, again, Madeline laid out, this idea of a march of progress, that there was linearity to human evolution, that a chimpanzee-like animal, a bent hip, bent kneed knuckle walker, slowly and surely evolved into a more upright human. And so dots were connected between these fossils, which is easy to do when you only have a couple. But in the last decade or two, there has been this extraordinary burst in the discovery of new fossil foot bones. And I've been so lucky to be studying feet at this time in the history of our science, when so many new foot bones have been discovered. And so here we have the foot fossil record, the hominin fossil record from the last 4.5 million years. And there's, again, just been this remarkable explosion in the number of fossils that we can work with to test hypotheses about foot evolution. And so that's what I did with my colleagues a couple of years ago. My graduate student, Ellie McNutt, who's now a professor at Ohio University, and my good friend Bernd Zipfel, who's in South Africa. He was a podiatrist early in his career. And as he puts it, he got sick of fixing people's bunions. And so he was interested in fossils, and he went back to school and became a paleoanthropologist, but he's got this expertise in the foot. And what we did is we met at Dartmouth, and we had 3D scans, and we had casts of all of or many of the hominin foot fossils, and we laid them out on a big table to try to make sense of what was happening. What is the pattern that we see over the course of foot evolution? So this table here shows you all of those fossils. And if you think of it as a graph, along the x-axis would be time from left to right, and on the y-axis would be the part of the foot. So the heel, and then the talus or the ankle, and then the medial cuneiform, all the way down to the toes. And what we realized when we laid all this stuff out was that foot evolution was super complicated. It was much more complicated than this simple linear narrative that has been the story of human evolution for a long time. It wasn't simple, and it wasn't linear. We then traveled all over Eastern and Southern Africa and visited these museums where the original fossils were kept. So that insert picture there is Ellie and Byrne in Ethiopia, looking at the Ardipithecus ramidus foot material. Okay, so as I mentioned a moment ago, humans and chimpanzees are sister taxa. We've got the same 26 foot bones in our feet, and they're just these subtle modifications. But to really know what's going on, you need the fossils. And so what we hypothesized was that we would see foot evolution progressing through these stages. Okay, and so here's what we imagined. Okay, so this is the hypothesis that we can test, is that you have from top to bottom a chimpanzee-like foot, an ape-like foot, generalized ape foot, doesn't have to be chimpanzee-like because they've evolved too, but an ape-like foot that then goes through a time period in which you still have the grasping big toe and Ardipithecus, but there are some hints of bipedalism. And then through an Australopithecus stage where it's much more human-like, but those feet still have long curved toes. And then you get to genus Homo, which is us, Homo sapiens, and our extinct cousins like Neanderthals, or something I'll mention in a moment, Homo naledi. Okay, so there would be these three clusters. So what we did was we took all of the foot bones and we measured them, and we made all sorts of observations on their characteristics, and we put them into essentially a family tree to see, do they cluster in this way? And the answer is no, they don't. In fact, they cluster in this absolute mess. And so I'm not expecting you to understand what I've just put up on the screen. I just want you to see that it's a mess, that you don't have these clear, big, defined clusters. So let's find humans. Humans are at the very bottom. Okay, there we are. And we do cluster with other members of our genus Homo, but also with an Australopithecus, with Lucy species. So where's the rest of the Australopithecus? Well, okay, they're kind of up here. They're scattered up here. But then we have all of these earlier hominins that are arranging themselves like spokes on a wheel rather than clustering together. And so what this says to me, the way I interpret this, is that early in hominin evolution, terrestrial bipedal evolution, there was experimentation. As Per said, there was a radiation. There were lots of different forms of bipedalism. There were different feet that would have been adapted for different landscapes and different combinations of moving on the ground and climbing trees. And so we think that there is experimentation going on early in, at least in Africa, with terrestrial bipedal origins. But then later on in human evolution, what we see at the time of Australopithecus is again experimentation. And the best way I can communicate this is by telling you about two different fossil taxa. One is Australopithecus afarensis. This is Lucy species, known from Ethiopia and sites in Tanzania. And it dates from about 3 to about 3.7 million years ago. A much more recent species, geologically and also historically, discovered in 2009, just under 2 million years old, is Australopithecus sediba, known from a cave site in South Africa. Now, given that sediba is 1 million years younger than Lucy and her kind, one of the expectations might be it would be more human-like. Okay, so let's have a look at the evidence. So here's Lucy and another skeleton of an Australopithecus afarensis found by Johannes Haile Selassie in the Woronzo Mille area of Ethiopia. It's known as Katanumu, or big guy. So we have these beautiful skeletons. And then in addition to that, we have fossil foot bones. And we not only have fossil foot bones, but we have fossil feet from a juvenile, the Dakika child shown on the far left. There are the Laetoli footprints measured by Per. And then the skeletons allow us to reconstruct gait with some degree of specificity in Australopithecus afarensis. And though it's not exactly like gait in humans today, it is very, very human-like in how they moved. Okay, so if we move now to Australopithecus sediba, 1 million years later in time, again, the expectation would be that it would be at least as human-like. And what we found instead is that it's not. It's not at all. There's a combination of anatomies in these skeletons that are entirely unexpected. The ankle joint in some ways is less human-like than Lucy's. The mid portion of the foot is certainly less human-like. And the heel is extraordinary. So I'll focus in on the heel for a moment. In the top left is a human heel looking at it from the side. It looks like a big potato. It's a big chunk of bone that is useful for, adapted for distributing forces across the bone as we heel strike in our bipedal gait. Lucy species, shown in the upper right, same sort of anatomy. There's subtle differences, but in general, this is a big, chunky bone adapted for heel striking bipedalism. Bottom left is a chimpanzee. Bottom right is Australopithecus sediba, looking much more chimpanzee-like than any of us would have expected for this time period in an Australopithecus. And we've done all sort of the fancy geometric morphometrics on this, and sure enough, Afarensis plots with humans and Sediba plots more like what we see in the apes. And so I've been working with a team trying to figure out, well, what does this mean? And what we have reconstructed for the species is a gait that is different than what we see in Afarensis. It's what we would call a hyperpronation. If we combine together the elements of the foot and the ankle and the knee and the lower back and the pelvis, we get a species walking on two legs, sure, but walking in a way that's different from what we see in other species of Australopithecus. When humans do this today, because some humans do hyperpronate, we have all sorts of problems with our back and our hips and our knees and our feet. It's a pathological gait. The skeletons that we unearthed in South Africa have anatomical solutions to the very problems that humans have if they walk this way. And so we think that they're adapted for this form of bipedal gait. So the idea here is that different species of Australopithecus would have been walking in different ways. And I'll end with the fact that it's not just Australopithecus. If we jump now to more recent time periods, the Middle Pleistocene into the Late Pleistocene, again, it's not just Homo sapiens. There are Neanderthals, Denisovans, Homo floresiensis, this really peculiar island population on the in Flores. And then there's a relatively new discovery, again from South Africa, of what's called Homo naledi. And I've been working on Homo naledi. The beautiful thing here is just the quantity of fossils we can work with. There are literally thousands of fossils found in a cave called Rising Star Cave in South Africa. And we have been attempting to work on gait reconstruction. And what we found is that this species probably had a pretty flat arch, didn't have a nice, well-developed arch like humans today. And it had very, very small joint area. Its knees and its hips and its ankles were very small, which was strange to us. And we think what it means is that it doesn't have, the species wouldn't have had the adaptations necessary, skeletal adaptations, to have a large home range. And they actually would have not been able to walk across great distances like Homo sapiens can. And so again, as we go into the Pleistocene, we see the same sort of thing of different species coexisting with each other, different populations, because we know that some of them exchange genes with each other, like humans and Neanderthals, but that they walked in these slightly different ways. So bipedal experimentation happening throughout human evolution, which is in contrast to that very simple narrative, that simple imagery that Madeline showed, that we see on coffee cups and t-shirts and bumper stickers of that chimp slowly turning into the human. It was much more complicated and frankly, much more interesting than that. So thank you all very much. I really appreciate the organizers and the collaborators. And then for anyone interested in bipedalism, I have a shameless self-promotion that I have a book coming out on April 6th called First Steps, How Upright Walking Made Us Human. Thank you.
Jerry, thank you very much for your insights in the walk of life. Watching your graphic, at least for me, I think the human evolution is really much more complicated than at least I thought. Now we're coming to our next speaker. Our next speaker is Sarah Elton from Durham University. Sarah focuses with her research on the variation of old world monkeys. Old world monkeys that evolved around the same time as many human ancestors, and old world monkeys that were found in the same places as many human ancestors. The knowledge of these primates give us a comparative context for human evolution. Sarah, the title of your talk is Did it happen by chance? Neutral models in primate evolution. Sarah, please.
Thank you very much, Oliver, and thank you very much, everyone, for your fantastic talks. I'm just going to try and share my screen and put my slideshow on. So just bear with me. Lovely. Hopefully you can see that. Is that okay?
Perfect.
Great. Thank you very much indeed for organising this. There are lots of things that we don't like about lockdowns, which include the fact that you can't have your hair cut for several months, but this is not one of them. It's fantastic to be with all of these brilliant people talking about very, very interesting things. And I'm going to talk about something that's slightly different. So I'm going to pick up mainly on other parts of the primates fossil record, so talk about the monkeys, but also think about some of the similar themes in terms of diversity and try to think about how we might examine the material a bit differently. So I'm going to think about neutral models in primate evolution. So anthropologists, paleoanthropologists are often trained in very classical neo-Darwinian methods. So we think about selection, we think about adaptation, and we don't often think very much about processes such as drift and random and neutral factors that may cause evolutionary change. Now, equally, many of us think about ecology, and you've seen some absolutely beautiful work today, thinking about not only the morphology and the skeletal adaptations of hominins, but also think about where they were living and what they were doing. And within ecology, there is a movement to also think about neutral models. And so, there's a really lovely piece of work called the Unified Neutral Theory of Biodiversity and Biogeography, where you think about, well, is everything that we think about in terms of evolutionary ecology, is it adaptive? What about stochastic models? What about things that happened by chance? And some of the things that we're interested in human evolution are about the number of species, how they're distributed in space and time. So thinking about biodiversity and biogeography, and we've seen some good examples of that today. Also what species and individuals do, where they live, what they do, who or what they interact with. And these are the things that we think of in paleoecology and paleobiology. Another thing that we're very interested in is what is the context for human evolution? So what are other species doing in the same times and places as hominins, our closest relatives? So ecological neutral theory is a really, really interesting theory to try and mix it up a little bit, think a bit differently about the material we have and put it in a framework that doesn't fall back on traditional explanations such as niche partitioning, adaptation, but think about things that may happen by chance. So on the slide here at the top left, really lovely piece of work about niche partitioning in neotropical primates, so modern primates found in South America. And there's been a lot of detailed work here and found that the primates there live in different parts of the canopy, they eat different things. So this seems to be a really good model for niche partitioning. So when we talk about ecological neutral theory, we're not necessarily saying that this explains absolutely everything, but this may explain some of the things that previously we found to be quite difficult to contemplate using traditional models. And I'll come on to that in an example from Southern Africa in a moment. So within ecological neutral theory, we're thinking about chance factors. So chance deaths, chance births, chance immigration, that creates taxonomic diversity rather than taxonomic diversity that's created through an ecological niche opening up and species filling that. So, you don't necessarily, under this neutral model, need to have animals eating different things to avoid competition. You may have diversity appearing because of these very chance factors. Now, I first started to think about neutral models quite a long time ago, when there were some things I really was struggling to explain in the monkey fossil record from Southern Africa. So let me take you back to the early Pleistocene, to the cradle of humankind, which is in modern day South Africa. So you'll see on the slide here, the red square, which is the relatively small region, which is the UNESCO cradle. And this is this incredibly diverse fossiliferous region that Jeremy was talking about in the context of, for example, Australopithecus sativa and Malapa. And you can see that is one of the sites on here. I'm just going to use my mouse here, just there. And so when we have a look at the cradle of humankind, it's quite a small region, 470 square kilometres. And this includes two of the other famous Southern African Pleistocene sites, Taung and Makapanskat, that are further out. So the monkey diversity from the cradle is really quite remarkable. We've got these sites there where you literally can stand there. If you can stand at Kromdraai and look over to Swartkrans, and it's an easy short walk from one to the other, yet they have very different species. There's also lots and lots of species that you find in that area. So I'm looking at this and I was thinking, they're all quite similar in terms of their adaptation. I'll show you some work where we've actually tried to quantify that in a moment. So how did they exist in this one place? Now, of course, some of this is because of time averaging. So these wouldn't have all existed at the same time. But even taking that into consideration, there does seem to be quite high diversity. So one of the things I'm going to look at now is, do we have a lot of monkeys in a small area? So these are just some simple graphs of taxonomic diversity. And there are various ways that you can measure taxonomic diversity. I'm using Meinik's index here, which is a fairly simple way, where you take the number of species and look at that in relation to the sample size. And you can see from this that there's quite a lot of richness in this sample. So ignore this one here, that's a single species from a single site. That's a Malappa again, and that's Papillon and Gusticeps. But basically, we've got a reasonable amount of diversity here. And this diversity doesn't change over time. So the sites on this graph are in chronological order, from earliest to latest. And the diversity really doesn't alter over that period of time. And that period of time, we're talking about over 1 million years. So just to put this into the context of other big primary consumers, and so we think about the other mammals that live alongside these monkeys in Southern African sites, including hominins. This is their taxonomic diversity. Again, reasonably diverse. The diversity goes down over time, which is quite interesting. We don't see that happening in the monkeys. Now let's put these data into a slightly different context. Let's think about them in terms of modern monkey data, diversity data. So we take our modern game parks and look at old world monkey species count. So this is just really a really simple illustration. And we need to do more sophisticated work on this, but it does for the moment. So we have forest parks in Asia, forest parks in Africa, open parks and game reserves in Asia, and open parks and game reserves in Africa. And you can see the average areas there and also the species count. And compared to modern African open habitat parks, South African Pleistocene monkey species counts are higher in larger parks than the cradle. And the cradle size is equivalent to Asian open parks. And so what we can conclude here is that you've actually got quite high diversity in the cradle, particularly for an African assemblage. So one question is, are we managing to maintain this high diversity through partitioning niches? So let's have a think about that. So let's have a look at adaptive diversity in these prior Pleistocene monkey communities. And so what I've done here is just done a really simple quantification of locomotion and diet, and looking at the amount of diversity in each of these sites, again, on the graph from earliest to most recent. And the first thing, before we even get onto what they're eating and how they're moving, is they've all got really large body masses. Now, they've got large body masses in comparison to modern old world monkeys, where the mean phylogenetically corrected body mass is around about 6 to 8 kilos. Here we've got big monkeys. So these are the size of modern baboons. So a decent dog size, basically, we have. And so there's an awful lot of large monkey diversity. So we don't have lots and lots of species with lots of different body sizes. They're all big body mass creatures. And then when we have a look at the adaptive diversity, we really don't have very much richness at all. And again, that richness doesn't vary over time. So similarly, taxonomic diversity doesn't vary, but also our richness in terms of locomotion and diet. So locomotion, very dominated by largely terrestrial forms, and diet, a mixture of fruit and leaves, the odd bit of grass in Therapithecus, but there's not really any evidence at all here for niche partitioning, which is really, really interesting. So there's a couple of things that we also need to think about. So of course, the effects of taxonomy, which I like to gloss over and mention at the end of the talk, but we can talk about it more later, is that of course we have space and time averaging here, which may be upsetting the signals. We need to think about the depositional processes by which these assemblages were associated and how they were accumulated. Now, Pickering and colleagues suggest that the cave sites in the cradle are sampling drier periods, which may mean that we've got a slightly skewed signal here. So we need to build in the effects of taphonomy. We also need to think about what diversity is being sampled. So we've got different types of ecological diversity. So we've got gamma diversity, which is all the diversity that we have. We've then got beta diversity, which is diversity between habitats. And then we've got alpha diversity within habitats. Now, it's difficult to tell from the information that we have, even when we study it in quite a lot of detail, what sort of diversity, whether we're sampling the diversity within a given habitat or whether we've got diversity that's between habitats. And again, that will alter the way in which we examine and interpret the material. Now, if we can calculate species evenness, this may help to determine things like ecosystem stability, going back to this idea that we might actually be sampling particular types of habitats. So there are lots of ways that we could try and use the data in new and creative ways to get at interesting issues. Now, when I started looking at these monkey data, I was thinking, well, there doesn't seem to be much evidence at all for partition issues. We've got very similar large body sizes. We've got very similar locomotor strategies that again link to this large body size, because terrestrial animals, terrestrial monkeys are more likely to be larger, or rather larger animals are more likely to be terrestrial. And then we've got relatively little diversity in what they're eating. So let's make this baseline assumption then that the species that we have are ecologically identical. So in terms of body mass and trophic level, and also equally fit. So they've got an equal chance of surviving and reproducing, and their genes spreading throughout the next generation. So if we add into that these random stochastic effects, random death, speciation, extinction, dispersal, and this then causes ecological drift. And so what you get is you get this unstable coexistence between different forms, and you get this diversity. So what we may also have in these populations is soft neutrality. So where we have stochastic factors creating the diversity with some minor adaptive tweaks. Now, this is something that'd be really interesting to discuss, because a lot of the things that we look at in terms of diversity are related to the cranium. But when we look at the post-cranium, we may be seeing something different here as well. We may be seeing distinct adaptations to particular modes. And what we can see, for example, when we look at the Guenon postcranium, and Guenons are a group of African forest monkeys, you can see really quite distinct signatures in their skeletons that associate with different parts of canopy use, with different types of locomotion at the ends of branches and so on. So it could be that we just need to get a little bit better at looking at the terrestrial monkey postcranium to try and pick up very small adaptive differences. So that's something that would be really well worth exploring in the future. So how does this then apply to human evolution at a similar time? So I'm going to take Cubifora as an example. On the left-hand side of this slide, we have about 4 species at Cubifora. Over a time period, but you get quite high hominin diversity in certain members. So for example, a member might be 200,000 years long, however, but you still find these also associated for quite a long time, over time, and in slightly different spatial regions, but still in the overall catchment at Cubifora. And so we have multiple species of Homo, for example, and it looks like these are, they've got, generally, they're quite eclectic, they have quite varied diets, but there looks like there's a bit more niche partitioning here. So if we have a look at Paranthropus, and Paranthropus, as Jeremy said, is another form, this big heavy chewer, looks like it's doing something different with its diet. And this is not only from its morphological features, but also from its isotopes. So we may be getting something quite different where we don't have neutral effects going on in the ecology of cubivora hominids. However, we might have something quite neutral going on in Southern Africa, where we may have 2 or more forms of Australopithecus. And again, this is highly debated, and I'll leave that debate to the taxonomists, but it's possible that you got similar forms of hominin existing, not because they were occupying different niches, but just by chance, and because of these chance neutral factors. Worth looking at. So in the last minute or so, I'm going to talk about something else where we may use neutral models to examine things that are interested in the fossil record. And this is dispersal. So I'm just taking an example here of baboon dispersal. So what we have, if we look at the monkeys more generally from the Pleistocene, is we have a number of old world monkeys that are now extinct. So Therapithecus, Parapapio, Gorgopithecus, and Cercopithocoides that all exist in East and Southern Africa. And it looks like you probably got a southerly dispersal. So originating somewhere further north and then dispersing down into these cradle of humankind sites in Southern Africa. Now, the interesting thing is a really dominant member of the modern monkey fauna, Papio, is totally absent in Eastern Africa in the early Pleistocene. So we don't see it creeping into the fossil record, and then only in very small numbers until the middle Pleistocene. So what's going on? Because if we go to an African game park today, we see nothing but baboons. So I did this model, this dispersal extinction cladogenesis model, where you take modern species and you look to see what their dispersal patterns are likely to be. And it seems that Papio originated in the southern tropical region, so not quite down south where its fossil record is best known, but a bit further up in Tanzania, or areas around Tanzania, which is quite consistent with the information that we're getting from the molecular record. And so it appears to have been able to disperse south, but not disperse north into some of these really fossiliferous regions. So let's just look at cubivora. This is cubivora from the same period that I was talking about with the hominins. You see lots of monkey diversity, but very, very little Papio at all. So what is possibly happening here is you've got another neutral process called high density blocking at play, whereby this species columbine, so we see high columbine diversity. So all of these species here are columbines and terrestrial columbines. So we don't have a modern analog for those. It's likely that they are blocking, sorry, that's my timer going off, telling me to stop. It's likely that they are blocking the advancement of Papio. And also it's likely that Therapithecus is blocking the advancement of Papio into Eastern African sites. So rather than it being competitive exclusion, because these animals are not eating the same things as Papio do, Therapithecus eats grass, columbines are probably very leaf eaters, Papio is much more eclectic. It's just they filled the ecological space. They filled the space completely and Papio can't get down there. So then Homo disrupticus comes. So we have Homo agaster appearing in Eastern Africa, evolving in Eastern Africa. And it's likely that this has caused ecological disruption. So we see Therapithecus waldi on the left of the slide, exiting stage left, becoming extinct, possibly being hunted quite extensively by Homo. And then that then makes space for Papio to get out. The block is released and Papio gets out into Eastern Africa. And the rest is literally history. We know Papio lives very well alongside humans. At least it thinks it does. Humans often beg to differ. So in summary, it's likely that a mix of neutral and other processes, structured diversity in complex Pleistocene environments, but really more work is needed to formulate and test neutral models in primates and human evolution. Thank you.
Thank you very much, Sarah, for your insights and your talk. I'm really pleased to have 4 different speakers here with me. I didn't understand everything, but I tried hard. Now I want to remember the audience. Now you can post your questions in the Q&A window in Zoom to ask the speakers questions you have. I think it'll take you some time. That's the reason I want to ask all the four speakers. Maybe you have a question to one of your colleagues. Every one of you has at least one question. Maybe Madeleine, you want to start with a first question to one of your incredible colleagues. I think you have a lot, but maybe one question.
Oh yeah,
for starting.
Usually we exchange emails. Okay. Oh, yeah. I mean, I was really fascinated by all of your talks, and of course, many topics I know from your publications. So,
but what do you think the audience could be interested in as a general question?
Depending on the audience, if the audience are students on anthropology, they have probably a lot of specific questions. So
I think we pose a specific question because I think you have a very specialist audience today. I'm not a specialist, but I wonder always how with little fossils, for me it's just a bone. And you can tell stories about.
Okay,
you can write books on maybe one or two fossils.
Yes.
I wonder how you do that.
I mean, it's good that there are bones for you. For somebody, this is a stone. So
no, it's only bone because you're telling me that's a bone.
Yeah. No, I mean, what we are doing is comparative anatomy. So you have to know the anatomy of living species quite well before you're going to fossils. And actually that's why for us collections, museums are so important, and we are collecting also living fossils to have a comparative material. So this is the base. The base is the living world and of course the fossils, and then we compare them. And I have also, for those who are beginners in this part, to maybe advertising, I also write a book, Ancient Bones. It came out now in English. So if you are interested, you can read in these books, especially about methods, how we read the record. Not only the bone record, but also the sediment, the stone record, because time, which is so important for all the understandings about evolution, time is only represented in sediments, in stones. So you have to know how to read stones, how to read landscapes, to understand all these complicated things.
So, Madeleine, we have our first questions from landscapes to stones. This is a question for all speakers. Did dietary habitats of food play a role in human evolution? Maybe we start with Sarah. You had, I think, at least what I guess it was a topic of your speech on the habitat.
Yeah, thank you. Yes, I think it did. It has to. So one of the things that I was trying to do was be a bit of a disruptor and giving different models and different ideas and frameworks for how we can examine this. But when you have a look at the hominin fossil record, you can see really clear evidence of adaptation to different diets and different types of locomotion. Now, one of the questions I have for Jeremy and also for the other speakers was, how much difference do morphological, so when we look at morphological traits and we say, oh, well, you have you're overpronating here, you're hyperpronating, how much difference would that have? So is that related to adaptation, a very small adaptation to a particular substrate? Is that just something that just happens and then you get corresponding changes in the rest of the skeleton to accommodate it, and all of that sort of thing? So I think that we still have quite a lot of work to do to see, to try and closely link selective pressures to features in the skeleton and work out what is actually caused by selection and what is something that's more neutral, drift, occurs by chance, and then may be selected out or may be selected for later on. So I think that I'm going to pass over to Jerry, if that's okay, to try and answer that question about hyperpronation.
Sure. So it's something we struggle with. How do you interpret variation? And so when we find a fossil and compare it to others, they're never going to look identical. So when you see differences between one fossil and another, what does that mean? And so my starting point is always to look at that variation with a modern model in mind. And so given that I work with bipeds, I look a lot at human skeletons, of course, but you're also going to look at chimpanzees and gorillas and our other close living relatives to see, are the differences that we're seeing in these fossils, can they be encompassed within normal variation? But then because I'm a functional anatomist, what I care about is what do those differences mean in terms of function? And it's helped again to have modern models. And so one of the things we've done is we've had human participants walking on treadmills and walking on gait carpets, and then we will MRI them and look at their foot bones and see whether hypotheses that you have about functional anatomy, the foot as it pertains to gait, actually plays out. Or is the foot so functionally redundant? You know, with 26 parts, can't you add them up in different ways and actually get the same ultimate product? And so that's something that we've been playing with. And honestly, Sarah, you know, I don't have a good answer to your question. It's a difficult one, and it's one I'm constantly struggling with. But I sort of came into this looking at Australopithecus as pretty monolithic. When I finished my thesis, all the foot bones that I looked at in South Africa and Eastern Africa, an Australopithecus was an Australopithecus to me. They all walked in more or less a human-like way, with subtle differences. Sure, they could get into a tree better than I could, but this is a pretty good terrestrial biped. And so when Lee Berger found Australopithecus sediba and invited me to work on the foot bones, my response was, sure, you know, I'll take the measurements, I'll plug them into graphs I've already made, and we'll have a publication, you know, soon, right? And instead, these bones just floored me. I did not expect to see the anatomy that these bones contained. It was shocking. And instead of then trying to force them into a paradigm that I had created, it was, you know, maybe it was just early enough in my career that I could sort of see like, whoa, this is pretty cool. This is different than I would have expected. I'm going to have to write Contra de Silva a couple times in my publications, but that's okay. Changing your mind is actually a healthy thing to do when the evidence demands it. But you know, the final thing I'll say is that even my, you know, the interpretation I've now made about sediba is subject to change as we find new fossils. We could use some juveniles to see, you know, how do these traits develop? Do they develop over the course of the lifespan of the individual, or are these true genetic adaptations that these hominins are being born with? We don't know because we don't have the material yet, but that's why you keep digging.
Yeah.
Thank you, Per. I think Per wants to say something on it.
Yeah, I'd just like to follow on from this because this is what I found. Well, for a lot of things interesting about your talk, but a particular thing that really struck me was this, that we're going to see diversity among early hominin feet is no very great surprise, and that we are going to have a gradient, because there's an obvious trade-off between better climbing ability, in particular with a still grasping hallux, and you know, optimizing for terrestrial walking, and you get rid of the grasping function, and you might have sort of balance points on that. On that scale, as it were, from one end to another. But the thing that really intrigues me is the fact that you seem to have variability that's quite orthogonal to that is not, I mean, unless we're, you know, perhaps just not fully understanding how these feet are working, but it does seem to me that you have a number of foot morphology that are essentially about terrestrial locomotion, but just doing it differently, and that really intrigues me.
Yeah, me too. I think it's fascinating, you know, that they're biomechanically there are multiple ways to do this. But then fitting that within the framework of, you know, looking outside on my college campus and seeing that every human out there also walks a little bit differently, and so you know, interpreting those foot morphologies within that framework of, look, you know, there is bipedal variation that's going to exist within a species. And then at what point do you decide, do you see enough differences? And to me, it's got to be functionally important differences, where you then make that leap to, and these sorts of gaits are going to characterize the whole species. Yeah.
Thank you. We have another question. I think this question could be for Madeleine. Is the out of Africa hypothesis still valid, or lies the cradle of Homo in Europe, or even in the Allgäu? I think the Allgäu, this must be your question, where Udo was found.
Okay, I hear this question quite often. Then I ask back, what you mean with humans? What is humanity for you? Is the humanity Homo sapiens, the genus Homo maybe, or hominins? And this depends on the answer. We know that the oldest Homo is from Northern Africa. The oldest Homo sapiens is from Northern Africa, from Morocco. However, and then you see how difficult is the question, this is for me not Africa, that's for me Mediterranean, because it's north of the Atlas Mountains. So what is the answer for Homo sapiens? It's quite difficult, and every new find will change the question. For the genus Homo, it's clear that the oldest ones are Eastern African, so far. So we have not maybe to look in a political boundary, because actually the continents are political. So this is a heritage from the Roman time period. So the Romans looking from Rome and say in south is Africa and east is Asia and we are Europe. But this is an artificial boundary. More important are environments, climates and ecosystems. And if there is a suitable niche for a human, the human will occupy this niche. And later on, of course, you have cultural influences. But at the beginning, it's an ecological question. And for me, this is not an important, what is the birthplace or the cradle. So that's why I would not to say that it's Germany for the bipedalism. It's obviously not the case, because you find then another skeleton from Italy, and the things will change again.
Yeah, maybe such a question is more based on local patriotism. Well,
of course, but I understand local patriotism. Yeah. Everybody should love the local area, but this is not a scientific point.
Okay. So to the other speakers, do you have any questions to each other? Because there are no questions at the moment from the audience. You have to mute your mic, Sarah.
I have a question, if that's okay.
Yes, and then Cherry, the next question, but it's your turn now, Sarah.
So just in relation to when we look at the skeleton, how much of this, what we see, do you think is related to plasticity and what organisms were doing when they were growing up? You're
asking who?
Both Madeline and Jeremy, and Per as well, actually, because it's relevant to everyone, I think.
So maybe Cherry, you start.
Yeah, sure. So one of my favorite fossils that I've had a chance to work on is the Dakika foot, and it's from a juvenile Australopithecus afarensis, and that was exactly what we had to contend with. Were those questions of comparing it to the adult. What do we see in terms of developmental changes that are probably related to what the organism was doing when it was alive, versus these true sort of genetic adaptations. And one of the surprising things we found was that the heel bone of humans, when you're born, your heel is very chunky. It's already sort of ready for the rigors of bipedal locomotion, even though you won't walk for another year or so. And skeletons that I've seen of individuals who have never walked still have very large heels. They don't have much trabecular bone in them, but they're still voluminous. The Dakika child doesn't have that. So the juvenile looks like it has a tiny sort of thin chimpanzee-like heel that's not very large, but the adult looks like it has a very large one. And so I think they are developing their large heels in a different way than humans do. We also saw it with the toe, as Per pointed out. Australopithecus afarensis doesn't have a big toe in line with the other digit. It has a big toe in line with the other digits, doesn't have grasping ability, and the bones would reflect that. But the juvenile looks like it has a little more mobility to it. And so the kids, as kids today do, might have been climbing a little more frequently, but then as it got bigger and adopted more terrestrial bipedalism, it would have become a more rigid joint. And so that foot was so great in sort of helping us tease apart what features looked like they were telling us about the lifestyle of the animal versus which ones were true sort of genetic adaptations, or even, unfortunately, these phylogenetic inertia carryovers from an ancestor that was doing something way back, and is frustrating our ability to functionally interpret the fossils.
Do you want to also answer, or Madeleine?
Yeah. I can add a very interesting observation when I study foot bones from living great apes, from gorillas and from bonobos in collections and chimpanzees. At the beginning, I measured every individual simultaneously, and then I realized there is a difference between a zoo animal and a wild animal. And luckily, I had collections with a lot of wild-shot animals. And then I saw that if a zoo chimp did not climb in his life, then the foot bones looking different. And this is interesting for the plasticity. So that means certain characters, like the torsion of the big toe, so the torque, the big toe, is something which is plastic. And if you find it in the fossils, it's really indicate that the animal has used it exactly in this manner. And so that usage of this or if you don't use it, it is not genetically fixed. And this was for me a very interesting observation. So it shows that we should be a little bit careful if we see variance in some morphological details, because it can be plasticity. It can also be that the males and the females use several positional behaviors in a different frequency, and then you get a little bit different morphology.
Do you want to add, or pose a new question to one of your scientific colleagues?
Can I throw something out, actually? There's something I'd be interested in discussing with Jeremy. I realize that this is going to start getting potentially onto the picture of things you can't say about Laetoli, so fair enough. But as a general point, I'm interested in the question of detailed analysis of trace fossils and tying them in with the body fossil record. Now, of course, frequently we have problems about taxonomic attribution, but in relation to the Laetoli tracks, where you have, this is a nice data set. They're very three-dimensional. You've got rather a lot of footprints. Without scooping yourself, and I appreciate you being probably recorded and all this sort of thing, but can you tell me a bit generally about your thoughts here about integrating the body fossil record and the trace fossil record into a kind of cohesive analysis?
Yeah. No, it's and I so appreciated how you walked us through the those footprints on Crete, and how oftentimes, certain aspects of the footprint can tell you about the general shape of the foot, and other times it's telling you about substrate, and other times it's telling you about, that interplay between the two. And you're right that at Laetoli, I think one of the real gifts of Laetoli is the number of footprints there are. And so you can begin to see places where the prints are giving you good anatomical detail versus places where, that's just a sloppy print, that's substrate shifting.
Yeah.
And how easily it could be that you would misinterpret that as, for instance, there's one footprint that has been misinterpreted as having more of a divergent big toe, when my understanding of it is it's just a raindrop in that spot.
Sure.
And so, having as many of them as possible is going to help with that. But I still think you have to interpret it with real caution.
Oh, yeah.
And what I really like is work Kevin Hatala has done, he's at Chatham University, he's done some really lovely work using, testing some hypotheses of function of the foot by taking the actual volcanic ash from, this is a site in Kenya called the Illoret site, and wetting it down in the same way that they've been able to reconstruct what the sediment would have been like at that time, and then having people walk through that. And collecting all sorts of kinematic data on people as they're then leaving the footprints. And testing relationships between variation in the footprints that they get and variation in flexion of the knee, extension of the hip, just basic aspects of gait. And so if you don't then incorporate in, you know, the substrate that they're walking in, I think you run into some trouble. So I think with Laetoli at least, to me, I've approached it more as sort of a bigger picture that from the footprints, it looks like there is an incipient arch, but not a very well-developed arch, like you said, that the big toe is in line with the other digits, that the digits look a little longer than you would expect for a human of that size, but that the forefoot is, I'm sorry, the hind foot is quite elongated. And to me, all of those features match really nicely, independently what I see in the bones from Hadar. Indeed. And so to me, those footprints match Australopithecus afarensis. But we know that there were other critters on the landscape. And so, you know, it wouldn't be surprising then to find footprints that don't quite match the Laetoli ones. But then the question is always going to be, you know, is it because of substrate differences? Is it because of pace differences? Is it because one's a child, one's an adult? You know, or just individual differences. We're going to be constantly fighting with each other over, again, how do you explain variation?
Yeah, exactly. But I think these points are really important. If nobody minds, I'd like to do a screen share again, because there's an image here that's kind of useful. You know, I obviously skated over this stuff in fairly quickly with a 15-minute slot. But what I want to draw your attention to with this particular print is the slight oddities about it. If you think about this in terms of a human, I mean, all right, so you've got a ball here, but you've got an individuated expulsion rim around this part. If you just step flat down, that wouldn't happen. You wouldn't get this edge here. And there's a sense of the whole thing having sort of drilled down into the sediment. This expulsion rim is a little bit too emphatic. And the alignment of the digits, if you think about it, is odd, because this is the long axis of the foot. And you would expect the digits to align neatly with that, as you'd see in, you know, there's actually my daughter's foot there, handling for comparison. In fact, they are, as you can see, deflected clockwise by of the order of about 10 degrees or so. So I have to say, I once performed some experiments with myself and my foot in a sandbox to try to replicate this. I don't have a picture, but I think essentially what is going on is that there's a stride here. You know, the guy stepped down, but when it's rolling onto its forefoot, and now it's pivoting to the right, and the ball is actually grinding into the sediment here, and by the time the toes leave their impressions, if you imagine it's coming into he or she is coming into the stride in this direction, but is leaving this direction. There's a little bit of a twist built in, and this comes back to what I was saying about, you know, you can't look at this and imagine that this is a cast of the foot. It's
right.
It's like a little film clip in stone created by the interaction between the foot and the sediment. So one has to be really careful with the interpretation. And if you look at some of the others from the same, I mean, like A here. Oh, go away, Robinson. Let me see if I can find. I want to see if I can get that other little foot up without Robinson Crusoe on it. There we go, much better. If you look at A here, so a rather smaller foot, you can use the same scale bar. You don't have that twist. This is a straight step, and you can see from the alignment of the toenail drag that it's going straight forwards. C here is another matter. Again, something really interesting going on with the shape of the hallux, which possibly has actually been is actually rotated here. You can perhaps see, especially in the color-coded image, there's this very distinctive sort of boat-shaped concavo-convex outline, which is in fact what your big toe, or indeed your thumb, looks like in side view. And there's a skin crease here, which we don't get in the others. So, of course, what's frustrating with this is that we have a small data set. You know, we would wish to have many more tracks, and we have hopes for, you know, obviously we're going to be scouring the area for more. But with the best preservation of these, you begin to get hints of what these feet are actually doing, and it's a little more complicated than just walking around.
Thank you, Per. We are nearly finished with our time, so please allow me to pose you one last question. I think it's one question which bothers me a bit. I think you're at the top of the current research, and I want to know from you, is it more like that your findings are contradicting older, maybe less complicated theory, or are they completing the older theories? And if they are more or less contradicting older, less complicated theories, how is the discussion in the scientific community? Is it more competitive, or is it more, well, we want to know your finding is not right? Feel free to start answering.
So maybe I start, because the discovery of the new views has indeed created much noise, and I think the data, if we collect more data, not only if we investigate old fossils, but if we identify new fossils, find new fossils, if by fieldwork, we will create a more complex theory. That's normal. And in the case of the Nuvius, a new continent, again, Europe came in focus, and the new time period. So the middle to late Miocene, nobody has thinking about that this can be important for human evolution or for bipedal evolution, and it is. And that's why I think it's make the picture more complicated, but it's we coming near to a reality. We will never reach reality. We will never have the last answer, but we are trying. So our life is short, but we are doing our best.
Thank you. Anyone of you to add?
Yeah, I could pipe up, I suppose. I mean, I think I should perhaps speak specifically for my own work, although I think in a way there's more general truth here. I mean, we are, strictly speaking, I think, just doing what people have always done. Of course, we are in part contradicting what has been said before. It wouldn't be much in the way of science otherwise, but we are substantially nuancing the previous perspectives. If you look, for example, at what the Trachelose trackways say, it's that, you know, we would already have said before that, well, okay, in East Africa at this time we have a probably bipedal hominin aurorin, okay, and now we would appear to have tracks of a bipedal hominin in the southernmost Balkans. So strictly speaking, it becomes a rather minor biogeographical statement. What has troubled me in the discussion, and I feel there is perhaps a problem in the discussion climate within the paleoanthropological community here, is that the Trachelose trackways have met a great deal of hostility, and in particular, what has kind of surprised me is that a number of people have asserted that this is an absolutely extraordinary claim. It's not a completely banal claim. If you're talking about a group of antelopes or pigs or some other component of the Miocene mammal fauna and say, well, hey, you know, we had these guys in East Africa already, but look, we found one in the southern Balkans. The response would be on the lines of, well, that's rather interesting. It might even be worth publishing. It's not actually a drastic conclusion. I think there is an issue with people, some people, being very, very invested in particular interpretive models. And I think that does cause a problem in the way that the discussion actually proceeds, and perhaps makes it more difficult to incorporate this sort of slightly off the wall data and seeing whether you know the interpretive models need to be modified at a deeper level. That's what I think, anyhow.
Thank you. Sarah or Cherry?
Sarah, you want to go?
I was just going to say, I think Per's absolutely right. I think that when we work with the fossil record, we are doing science. So we need to keep an open mind, and we need to look at the evidence and what the evidence is telling us. And it doesn't mean we shouldn't test the evidence. So by saying, oh, this is an extraordinary claim, sometimes people are saying, oh yeah, I'm just not going to believe it for the sake of it, but give me the evidence that I can use to actually verify this. But I think that we have a real issue with the fossil record in that we often don't have very much material to work with, and it would be lovely just to go out and sample wherever we need to or run experiments on whatever we have, but we've got to work with what we have. And so I think sometimes we need to use different methodological tools. I think sometimes we need to use different theoretical frameworks. But above all, I think we do need to go and look at other disciplines. So for example, if we were looking at this, Per's footprints in a mammalian context, our minds wouldn't be blown so much, because we know that, for example, we have lots and lots of African monkeys that evolved in Africa that went out into Asia and Europe at about the same time. So we've got Mesopithecus, for example, and no one says, oh, next extraordinary claim that this columbine got out into Europe, and possibly that we then had species coming back in to Africa from them. So I think that paleoanthropology does need to keep a much more open mind about lots of these things. And I think that will really benefit our discipline and benefit our science and strengthen our ideas. And if we can take the personalities out of it a bit, that would be good. So I think it's great to have debate. Sometimes it gets very personal, which makes it quite difficult as well. So yes, my rant is now over. Jerry.
Thank you.
Yeah, no, I agree with what everyone has said. There's a colleague of mine who used to say that the intensity of an argument is inversely proportional to the amount of data you have. and so, with a fossil record as small as it is, but bear in mind that it only gets bigger. So as we go out and find more and more and more and more and more, we're gonna begin to, and I already see this with my colleagues, especially younger colleagues, that there's a dose of humility that's been dumped on the field in the last few decades. There are so many discoveries in paleoanthropology that have been made that none of us could have expected. Who expected Homo floresiensis or Homo naledi and Australopithecus sediba I mentioned, Danuvius, the Trachylus footprints, that these are all somewhat disruptive discoveries. And we should start getting used to that. And sure, we need to be informed skeptics. And when a new discovery is made, we should challenge it. And we should say, well, what is the evidence? And demonstrate to me that your interpretation is correct. I mean, that's how you do science. But at some point, I think we also need to proceed as though we don't have this all figured out. And there are going to be new discoveries that disrupt the simple narrative that has been constructed by past generations of researchers, and that's okay.
Thank you, every one of you, for your opinion on this point. We are nearly finishing our Matariki Lecture Series 2021 on human evolution, and now it's time for Lucy to say some finishing words, because Lucy is organizing this event from Durham. She's from the Matariki Network. Now I see you, Lucy, on screen, so it's yours now.
Thanks, Oliver. So yes, my name's Lucy Tuzinski and I'm the Matariki Network manager. I'm based at Durham University in the UK, but I actually act equally on behalf of all 7 Matariki Network partners to help plan and coordinate the many and varied activities of our network. I'm delighted to be here today for the first of our new regular Matariki Lecture Series events, which, as VP Scher said at the very start of the session, is an exciting development from the previously annual Matariki Lectures. And we plan to hold events like today's at regular intervals throughout the year, featuring a wide range of researchers from right across the network, covering lots of different topics. So please do keep a lookout for these. If you'd like to find out a little bit more about the Matariki Network and exactly what we do, or if you'd like to subscribe to our quarterly e-bulletin, then please do get in touch with me by email. I'll put my email address into the chat there so that you can get the spelling of it, because my surname's not easy to spell, I appreciate. And also you can visit our website, www.matarikinetwork.org, for more information. And if you're on Twitter, then our handle is @Matariki Network, so please give us a follow on there as well. The network has recently celebrated its 10th birthday, and we've produced a special anniversary report to document the highlights of our first decade. We've uploaded the digital version of this report onto the website, or if you get in touch with me, I'll be happy to send it to you direct. We're always keen to work with new colleagues in different areas to extend and improve upon what we do, so please do get in touch. Lastly, just before I close, I'd like to just thank everybody who's helped make today such a success. Thank you to all of our wonderful speakers for your fascinating presentations and participating so fully in the discussion session afterwards. Thank you to VP Share for delivering such a warm welcome to our event. And, you know, last but by no means least, a big thank you to you, the audience, for tuning in today and engaging with our panel. I hope that you enjoyed the presentations as much as I did, and that you've learned at least one new thing about human evolution. I know that I definitely have. And I'd also like to give a special mention to our host today, Oliver Häusler, and his colleagues at Campus TV in Tübingen. Without them, this event would not have been possible, or certainly not, it wouldn't have gone as smoothly as this. They've worked tirelessly behind the scenes to take care of all the technical aspects of the event, for which I, for one, am extremely grateful. So thank you, Oliver and his team, and back to you.
Thank you, Lucy. It was a pleasure working with you, especially because I know Madeline for 2 years now, and I'm still excited about your findings. And that's on all 4 speakers. I want to know more about human evolution, even though it's getting maybe even more complicated the more findings you have. I am curious what the next Matariki Lecture Series will be about. Lucy, I think you know more. But for now, this is it. I want to say thank you to everybody, and I want to say goodbye, and have a good time, and till the next time on the Matariki Lecture Network. Thank you very much to everybody.
Thank you.
Thank you, and vielen Dank.
Thank you.
Thank you.